Now I want to go over like the basic steps of mitosis and the cell cycle. And as I said earlier, a lot of this should be a review of like freshman bio. This is kind of a random slide. You don't really need to know it so much, but like 1882, appreciate like how long ago it was that we first started like observing having the ability to observe some of those stages of mitosis. In order to do that, we had to have dyes because like a cell is really just like transparent. If you just like try to put it on the microscope, if you don't dye it with something, you're not really gonna be able to see much because light has nothing to reflect off of. And it was Walther. I don't know if it's a Walter or Walther. You never know back in the 1800s, right? Fleming was the person that kind of developed those dyes, so you could begin to see those phases of mitosis. All right, so the cell cycle. So you got to know this diagram. As I said, you've seen this. You should have seen this multiple times throughout your biology to career. We'll start here in G1. In the G1 phase, this is part of a broader part of the cell cycle called interphase. So think of like sports, like intermission would be like the in-between time of like the action and stuff. Interphase, if the action is cell division, interphase is like the in-between time when the cell is not dividing. Now, what's a little bit different from like sports is that interphase is about 90% of the cell cycle. And you can kind of see that just with how much of it is blue, right? 3 parts of interphase, G1, S, and G2. So G1, well, in all of them, we're doing metabolic activity and growth. That's common to all 3 of those parts of interphase. So G1 isn't that unique of a phase, but what you do need to make note of is that it's unduplicated chromosomes. So here we haven't made a copy of all the chromosomes. So here, the chromosome number would be 2N. We have 46 chromosomes here. Okay? Then, like, think about this. If we needed to do S phase, which S stands for synthesis, we're going to make another copy of all the chromosomes. And the reason for that is if I were to just divide up these 46 chromosomes, I would get 2 daughter cells that have only 23 chromosomes. And we don't want that. We want the daughter cells to have the same amount of chromosomes, those 46 chromosomes. That's why we got to make another copy of it. So in S phase, we make another copy. So now we get to what's called 4N, or that's not what 46 times 2 is. We get 92 chromosomes. So 46 times 2. So you'll be 4N. You technically have 4 copies of all the chromosomes. Okay? But then, and then G2 phase, the main thing for G2 is preparation. I think of it like Gatorade G2. I don't know if that sticks with you, but like Gatorade like came out with their like I don't even know if it exists anymore, but they were like trying to like
sucks.
Yeah, it's like I don't know. It's like was G2 like
Lower sugar.
it's the low sugar one. Yeah, that's right. Yeah, they made like the G2 the low. So like I think you're preparing for your activities. It's like your preparation. Yeah, it had that like weird aspartame kind of taste to it. Yeah.
It was so bad.
I don't know. One of the flavors is actually pretty good. Like it was actually like.
Oh, the light blue.
You wouldn't say it out loud, but you're like, oh, it's actually kind of. What? Anyways, all right. So G2.
You're an alien.
In G2, we're preparing, and we're also. I want you to think checking. What do you think we're checking? What do we just do that we should probably check before we just divide the cell? I didn't see anything. Make sure you copy the DNA correctly, because as I said, if you just like divide the cell without checking that you copy it, it's like you writing an English paper and not proofreading your paper before you submit it, right? Like maybe you're gonna be okay, but like a lot of times you're gonna make maybe some mistakes here. So we wanna check it to make sure we didn't make any mistakes. And let's say we find a cell, we're like, oh wow, we made a mutation in a really serious gene. Then we're going to kick that cell out, and we'll send it to a phase called G0 or G0, which we'll talk about a little bit later. These would be cells that either had errors, or this is mostly cells that are just like, kind of like not dividing at all. Like they're just kind of just doing their normal stuff. Anyways, so checking happens in G2, and then the M phase. So then we're going to move into two parts. This is cell division here. So let me find a way to do this. These two phases here, that is cell division. So cell division is broken up into the M phase, which is mitosis, and then cytokinesis. Or I'm sorry, I said that wrong. Cell division is broken up into mitosis and cytokinesis, and they call all of that collectively the M phase. So cell division and the M phase is referring to both of those. Okay? Cytokinesis, we divide up the cytoplasm. Mitosis, we just divide up the chromosomes. Any questions on this? Oh, and then once we're done, now we go back to 2N and 46. Right? So we divide up all those chromosomes to reduce that number of that chromosome number back down to 46. So you get two cells, each with 46 chromosomes, that 2N number. Okay. This should just be words for everything I said. Think PP-MAT. I don't know if I told you this story, but like when I was teaching freshman bio, I was going over the phases of mitosis, and I was like, man, I don't really know how you can like maybe remember the order. And somebody goes, PP-MAT. And I thought it was just like a typical freshman, just like being in. I was like, that is not. Oh wait, that's actually really funny. So PP-MAT, prophase, prometaphase, metaphase, anaphase, telophase. That's the ordering of the phases in mitosis. And then you have cytokinesis where you divide up the cytoplasm. So let's run through them. Before I actually show you mitosis, G2 of interphase, that's the G2, that's when we're preparing. We're checking the DNA, make sure there's no errors. Notice here how there's a nuclear envelope around our nucleus. That's going to help enclose the nucleus. You also have, sometimes you can have more than one nucleoli in the nucleus. So like the nucleolus, this would be where you anybody remember what we did in nucleolus? Not as important for this unit, but like, we made something there. Luke, you know? You said we did in the first unit? Yeah. Yeah, we made something there though. Did you remember? Ribosome. Yeah, you make the ribosomes and the nucleolus. Anyways, notice how the DNA is kind of this bowl of spaghetti. It is uncondensed. That's a key word to know here. We haven't condensed down that DNA because we're using the DNA. We're doing, we're making proteins here, right? We're using that DNA to make proteins. So then we're going to have to condense that DNA before we start, I was going to say cellular respiration, cell division. Some other vocabulary. You have two centrosomes. So you have one centrosome, another centrosome. So the centrosome, that is referring to, if I do it a little bit, if I draw them a little bigger here, you have these tube things that come at like 90-degree angles. The whole thing, like this whole thing, is a centrosome. But then if I'm referring to like one of these individual tube things, that is a centriole. So within one centrosome, you have a centriole pair, two centrioles, and a little less important, but they come at a 90-degree angle to each other. That's my attempt at doing that, like, you know, when you got like, you know, it's like that's 90 degrees. Okay. So during G2, we're gonna make sure we have two of those centrosomes because this is where you can sharpen this one. This is where we get the microtubules. So those little like lines there, those are the microtubules beginning to kind of like, they'll eventually come out of there, and we're going to use this to divide up the chromosomes. Okay. And again, the DNA number, we're at 2N, or 4N, I'm sorry, 4N. We're at 92 chromosomes here. Okay. So now we're at prophase. So now we're officially at the start of PP-MAT. Officially at the start of mitosis. So in prophase, you'll notice how the DNA, you can now see the chromosomes. So in prophase, you see the chromosomes, right? They're nice and condensed. That's a very, like, when you, like, they show you cells, like, is this cell in, how you know a cell's in prophase? If I show you the cell, you know it's in prophase. You see the chromosomes condensed, and you see the nucleus. Okay? So you see the nucleus, and the chromosomes are condensed. That's you. If you remember those two things, you'll know it's in prophase.
Wait, what do they look like uncondensed?
What's that?
What do they look like
Uncondensed.
uncondensed?
It's like a bowl of spaghetti.
Oh, okay.
Yeah, that's like it's all kind of stringy and stuff.
Or-
You'll then the aster. Aster is referring to like a star. And you know, like when you draw a star, like this is embarrassing.
Bruh.
That honestly might be the best star I've ever drawn in my life.
Patrick.
That's I'm pretty proud of myself right now. I got like a B in art when I was in elementary school, which I think is like impossible.
That's
Like I don't
crazy.
like. Anyways, aster means star. Like the arms of the star are the microtubules. The microtubules, and the microtubules, those make up the mitotic spindle. So then notice how the centrosomes are moving to opposite ends of the cell, and we're eventually going to use those microtubules, that mitotic spindle, to pull apart the chromosomes. You start seeing the centromere. The centromere is going to be like where we end up attaching the microtubules to pull apart those chromosomes. The nucleoli disappears. That isn't quite as important, but it technically happens. Yeah, that's going to be kind of, most of what you're going to get from prophase. Now going to the other P in PP-MAT, prometaphase. Now what you'll notice in and sometimes if you go over mitosis, they just go prophase, metaphase, but it's kind of like, like the people who like get super serious about this stuff, they're like, well, we're going to make a pro-metaphase, where we're not metaphase, like, let me show you metaphase. Metaphase, the chromosomes are meta at the middle of the cell. They're in the middle of the cell. Pro-metaphase, they're like moving there. Oh, I forgot to mention with prophase, like a prologue of a book, it's the beginning of the book. So prophase is the beginning of mitosis. All right, prometaphase, we're beginning to migrate those chromosomes to the center of the cell. You notice the fragments of the nuclear envelope. So the nuclear envelope broke down. Here you have the kinetochore microtubules. Those are the microtubules that actually connect at the centromere. So think centromere. They're the ones that are actually hooking up at the centromere of those chromosomes. The non-kinetochore microtubules, their role is more to like keep extending the cell. Extending the cell. Right? Because we want to extend the cell so we can break it up into two new cells. Notice how the centrosomes are on opposite poles here, there. All right, now metaphase. They're meeting in the middle of the cell. So there's your chromosomes lined up in the middle of the cell. They line up at something called the metaphase plate. Centrosomes are at opposite ends of the poles, and then, you see your microtubules. They're attached. Now, anaphase, A for away. A for away, or if that doesn't do it for you, check this out. You have an A right here, and it's separated by an N. Because in anaphase, we're separating the chromosomes. You know what I'm talking about?
Okay.
Yeah. So there you go. So what's going on? The microtubules are continuing to push. We're like pushing and pulling apart the chromosomes and also extending that cell so it's easier for it to break into two. Remember, I said like when you have your sister chromatids, they're technically held together by these cohesins all along the length of the arms. So here they're saying that, where is that? We're breaking apart those cohesins. We have to like break that down so that you can also pull apart those chromosomes. So like you have to remove the cohesins and you got to then pull apart the chromosomes. Not quite as important, but technically it's something that's happening. All right. Finishing up, so the last phase, so PP-MAT, And then T, the last phase of mitosis is telophase or telophase. I've heard it said both ways. Here, you now begin to see the new nuclear envelope forming. So you start to see that nucleus forming. It's pretty easy to tell a cell that's in telophase because you see two new nuclei forming. And notice how you haven't divided the cell yet. So a very common gotcha question is in telophase, you don't have two new cells. You have done, I said telophase, in mitosis you do nuclear division, right? We have divided the nucleus. Now cytokinesis, so the reason why they're both on the same slide here is because cytokinesis is kind of happening simultaneously. Cytoplasm, cytokinesis, we're pinching off that cell almost like a soap bubble. Now, there's two, plants do it one way, animals do it another way. This is showing an animal cell. So in animal cells, you form something called a cleavage furrow. In plants, they do something called the cell, what am I writing? A cell plate. Why would animals do cell division, cytokinesis differently than plant cells? What's the difference there? Catherine? The cell wall, yeah. So because plant cells have a cell wall, they have a cell plate, which is basically like a new cell wall forming. All right, animals have a cleavage furrow, and that's referring to kind of what's going on right there. Here's how you can remember it. Animals have cleavage. Plants don't. I don't think, at least. Maybe there's a version of cleavage in plants. I'm not sure. But anyways, plants, they have a cleavage furrow. They like to ask questions like that. You gotta find some way to remember it. Can you imagine saying that to a bunch of 7th graders? I legit said that, but just nobody looked up for the rest of class. Okay. Here's one slide combining everything on it. So I know it's kind of hard to read, but if you want to kind of one kind of tidy slide showing you everything, there you have it. So that's all I want to go over today. Let me stop the recording.